Está ligado. Tá. Okay, yeah. Okay, so let's try and start the last session of the day, and I'm going to talk about. I'm going to give sort of just a very quick tour through some general databases and programs that you can find online to study protein sequences and structures. And I mean, maybe some of you, I'm not gonna, there's like literally hundreds of these sites, and I'm gonna talk about maybe 5 or 6 of them, okay? And there's just some that I use a lot, so I can talk about them. And maybe a lot of you already know them. But I just wanted to, this diagram here at the beginning basically says, okay, if I'm going to study a gene or a protein, I can look up papers in the literature using PubMed. I can look up the sequence of the gene. I can find the position of the gene in the chromosome if the genome of the organism that I'm studying has been sequenced. I can find that gene in the chromosome. I can convert the gene into its protein sequence. Like I can translate the sequence of the DNA into the RNA, the DNA into the protein sequence. From the sequence, I can calculate the number of amino acids, the molecular weight, the theoretical isoelectric point. That is it a basic protein? Is it an acidic protein? Etc. That can be helpful if I'm trying to purify the protein. It's going to tell me what experimental strategy I can use to actually purify it. I can predict its secondary structure. Nowadays, we can just go straight to predicting its tertiary structure using AlphaFold and everything like that, which Fernando, I think, is going to talk about. Okay, but from the sequence, we can try and find other proteins that are similar. So the homologs, we can find homologs of a protein. So we can align the sequence of our protein with all other known sequences and find homologs. And maybe in these homologs, there might be parts of the protein that have been defined as domains. So I didn't talk about domains before, but like protein sequences, you have a sequence of a protein, and it folds up into its tertiary structure. But often, this tertiary structure might be sort of like this. Okay? There's the amino terminus and the C terminus here. Okay? And there might be like different parts of the protein that seem like they're as if they can sort of fold on their own, independent of the rest. And these are called domains. And often, but not all the time, but often, each domain may have a different function in the protein. They often work together. I mean, usually they're usually together for some reason, so they're working together. But they can be sometimes this domain can be found in combination with another domain in another protein. So it seems like evolution works at a whole bunch of different levels. Mutation of single amino acids, but also switching blocks of DNA from one gene to another. And often, not always, but sometimes, you can switch a block of DNA that codes for a certain domain and throw it attach it to the sequence of another protein. And now that other protein has an extra domain. And that extra domain might have another function. That extra domain now modifies the function of the protein, and that might be advantageous for the organism. And if it is advantageous, the organism will have more offspring. If it's not advantageous, or if it's detrimental, then that organism will die or not have that many offspring, etc. So over time, that gene can survive or not in the overall population. So that's domains. That's very, very important. So these homologs, you can align the homologs, and from this you can get information about which positions in the protein are more conserved than others. So there might be specific amino acids that are highly conserved, that never change, and there are some other regions that seem to be, they can change almost to anything. Okay, and you can use this kind of sequence alignment, and assuming that proteins with similar sequences are more proximal during evolution. That is, they have a common ancestor, a more recent common ancestor. You can use these alignments to make hypotheses about phylogeny or evolution, okay? And you can get, and nowadays, these large amounts of sequences allow us to make large sequence alignments and make huge, what they call homologous superfamilies, or superfamilies of homologs, that allow us to, that help us make models of three-dimensional structures of models of proteins. I'll talk a little about this later on in this talk, and I think Fernando's going to talk about this a lot in his talk later on. So this has the huge amount of sequences that have come about over the last years have allowed the production of algorithms that are much more successful in predicting three-dimensional structure, okay? And then you can use molecular graphics programs to analyze a model of the structure, okay? So what I'm going to do now is I'm going to sit at my computer and I'm going to try and just look at a couple of these sites, just to sort of like show you, like maybe a lot of you have already seen these sites. It's going to be boring. Maybe some of you may not have seen them. So let me see what I have here. So I had to choose a protein. So I chose a protein called glycogen phosphorylase, or sometimes just called phosphorylase. And it is an enzyme that we use when we're fasting. We haven't eaten in a while, and we have to start breaking down the glycogen, which is a polymer of glucose that's usually in our liver and in our muscles. We have to start breaking it down to maintain our glucose levels in our blood, okay? So this protein, it's a dimer, actually. I'm showing a monomer here. This is one subunit. It's usually a dimer. And it has an allosteric mechanism, just like I was mentioning in hemoglobin. And its activity is controlled at several different levels of ATP, AMP, glucose 6-phosphate, insulin inhibits it, adrenaline and glucagon activate it, etc. It's controlled at a whole bunch of different levels, okay? So, but let's say we're interested in this protein. And yeah, so it's involved in degradation of glycogen. So it hydrolyzes the glycosidic bonds between sugar molecules, okay? And it has a pyridoxal phosphate cofactor in its active site. So we're going to look at glycogen phosphorylase, glycogen phosphorylase from the muscle of rabbit glycogen phosphorylase, okay? So we're going to look at, there's a paper there's literally thousands of papers on glycogen phosphorylase in the literature. If you just get glycogen phosphorylase and put it in the PubMed site, so let me see if I have this here. Where am I? Where's my internet? Don't tell me I turned off my internet. No, I didn't. Okay. So, PubMed. So rabbit muscle phosphorylase. So this site here, PubMed, is a site for biological literature, okay? But you can also, I think it also has physics literature, all scientific literature, almost everything in here, okay? Biomedical, medical, chemistry, etc. Okay, so you can put in an author's name or a keyword or something. So if you put in rabbit muscle phosphorylase and put search, you're going to literally get 1,001 results. Okay, and then you can, and there are papers since 1954. You can see like there seems to have been a peak in research on this enzyme in the 1980s and 1970s. It seems to have tapered off, but there's still like, you know, many papers being published till 2024 at least here. Okay, so anyway, so I chose one of the papers, okay, which was this paper here, and it's a crystallographic structure. It's a paper relating the crystallographic structure of this protein. So anyway, but let's say I want to find the sequence, the gene sequence of this protein. So there's a site So if I go in the internet and I go to the NIH, the National Library of Medicine, okay, which is basically the NIH site, the NCBI site, sorry, which is called the National Center for Biotechnology Information. So this site is like a collection of a whole bunch of different databases. So it's got the PubMed database. It's got a whole bunch of databases of sequences of proteins and DNA and genomes and everything, okay? So if I just put in, what did I put in here? Yeah, if I select the gene, if I'm looking for the gene of rabbit muscle phosphorylase, then I will get to this page here. And here, it's basically showing the gene. This is the gene of the glycogen phosphorylase gene. And these are all of the exons, okay, in green. These green boxes are the exons. And in between are the introns, which are spliced out during the maturation of the messenger RNA. So this is like the messenger RNA, this is the gene, and then the messenger RNA in eukaryotes, there's all these introns that are removed during splicing, and then you get the mature mRNA that only has these green blocks here. But anyway, so if you put your mouse over this, there will open up, should open up. Yeah, now there's a whole bunch of links here, and they have this link here will give me the link to the nucleotide, sorry, the second one here will give me a link to the nucleotide sequence. So, of the mRNA. So it's downloaded a FASTA file. FASTA files are files that have this format. So FASTA files will have a format of the sequence of the DNA or the protein in one letter code, and always the first line of these FASTA files has like a little arrow and then like any kind of name, like some kind of label for the sequence. And then starting from the second line, it's just one letter code all the way down. In this case, it's a nucleotide sequence, so there's 4 different letters. Okay, and this is the mRNA. This is the messenger RNA. It's the mature RNA. It's lacking the introns. Only the exons are here. Okay, so I can, if I don't know what the sequence of the DNA is, but I have the mRNA sequence, okay, I can theoretically translate this sequence. Now, this sequence is already translated, in fact, on this site. If I go here, back here, I can get just the protein sequence. If I go here, NP, the other one was NM for mRNA, and this is MP, NP for protein. So if I click on this, it's going to download another file, and I get the protein FASTA file. Let me see, where is it? So here's the sequence of the protein with the one-letter code for the amino acids. It's also a FASTA file. It has the first line that has the code for the deposit. Okay? Anyway, so I can get either of these sequences. I can cut and paste them in subsequent analyses. But if I only had the mRNA sequence and I wanted to translate this, I could get the sequence. So I'm going to get everything except for the first line. I'm going to copy it. Okay? And then I'm going to go to another place, which is this IQUSP translate tool. So this IQUSP is a site. What does IQUSP actually mean? I can't even remember. Let me see here. It's IQUSP. It's like some kind of site. It's like Swiss Prot or something like that. I can't. No, I should know this. I can't remember. Home. Let me see what's going on. No? It's like a collection of a whole bunch of different programs. It's got literally hundreds of programs here for protein-protein interactions, lipids, whole bunch of things down here. You can explore it. It's gotten to be much more complex than when I used to use it as a student, and I only had a few tools. It was very, very easy. Now there's just so many things here, I get lost, okay? But you can explore this. But anyway, you put the sequence here. So I pasted the sequence, and now I'm going to translate the sequence. So translating it, and now here it translated it. I could have said translated only in one frame or everything like that. But basically here, it's translated the sequence in all 6 reading frames. Because you remember the DNA code, DNA is a double helix, and that there's strands going in opposite directions. So there's actually 2 ways to read the sequence. You can read it on one strand, or you can read it in the opposite direction on the other strand. Always in the 5' to 3' direction. But anyway, so you can, and there's 3 different reading frames because the codons are triplets. So you can start at position 1, 2, or 3 in the sequence that I gave it. So this is the 5' to 3' sequence from frame 1, frame 2, and frame 3. Seems like frame 3 is coding for a protein starting here. Oops, what did I do? Something wrong. It's starting from a protein here and ending here. So this is the full-length mRNA. So it had a sequence. It had some sequence of nucleotides that didn't code for anything because the mRNA will start at a nucleotide, and usually there's several nucleotides before the ribosome binding site and the first initiation codon. So here's the first initiation codon, is this methionine, and then it goes all the way to the stop codon here. And then you have the other coding reading frames in the opposite direction. But this is the actual sequence, okay? And actually, I can like confirm this. You can see this starts with MSR and ends in KIP. And if I look at the actual sequence of the protein, which I had downloaded here, it starts with MSP and ends in KIP. So that is actually the sequence of the protein, okay? So I can translate it, and then I could get this sequence. That could be the sequence of the protein that I'm interested in, okay? So you can get to the sequence of the protein in a whole bunch of different ways. You can go straight to also you can just go, instead of going to gene here, you can go to protein, okay? And if you go to protein instead of gene, you're going to go to the sequence, you're going to go directly to the site of the sequence of the protein. So I put in glycogen phosphorylase rabbit muscle, and I put in protein, and it gives me these whole bunch of glycogen phosphorylase muscle proteins. There's 200 of them because as well as the rabbit, this is the Latin form for rabbit, okay? It's also giving others, probably giving from other animals as well. If you keep on going down, you'll start getting the sequence from come from other animals probably as well, okay? And it's been deposited many, many times, probably because different labs have studied rabbit muscle gene expression, and they've deposited the sequences. So the sequence is appearing over and over again. It's the same sequence over and over again, okay? But anyway, so I go here, and I can get, it gives me the reference of where this sequence came from. Okay, there's a paper. And then down here we have the sequence. But here the sequence has these numbers, like it's giving me the numbers of the amino acids and everything. That might not be that, the format might not be that good. If I want to get the FASTA format, I go up here, FASTA, and I can get the sequence in the FASTA format. So I can copy this here and paste it and everything. Okay, so now I have the protein sequence. So there's several things I might want to do with the protein sequence. One thing is if I'm trying to purify the protein and I want to know the molecular weight or the isoelectric point or the number of, if it has cysteine residues or if it has a lot of acidic residues or basic residues or tryptophans, if I'm doing fluorescence experiments, like innate fluorescence experiments, I want to know how many tryptophans there are. I can take this and I can get this other site on XPZ, which is called protein parameters, okay? So if I go back here, protein parameters, and just paste in the sequence of the protein, okay? And this is just XPZ protparam, okay? And I go compute parameters. It gives me the sequence first. This is the sequence that I gave it. And then it gives me the number of amino acids. It gives me the molecular weight, the theoretical molecular weight, assuming there's no post-translational modifications, there's no disulfide bonds or anything like that. This is the theoretical isoelectric point. So it tells me that it seems like it's a neutral protein. The pi is around 7, okay? That means it has more or less an equal number of positively and negatively charged residues. You can see here it shows the composition of amino acids. So it's how many, there's 62 alanines, 7.4%, etc. Okay? And you can see here, yeah, there's 114 acidic residues, and there's 111 arginines and lysines. So the equal number of positive and negatively charged. And how many histidines are? There's 22 histidines, but histidines they have a pKa of around 6 or 7. So at physiological pH, half of them are going to be positively charged and half of them are going to be neutral. So that's why this protein has a pi of around 6.7 or something like that, okay? This is the atomic composition, formula, number of atoms, 13,682 atoms. Oh, and it's got the extinction coefficient. This is very, very useful if you're purifying the protein and you want to determine the concentration by ultraviolet absorption. You can use this extinction coefficient. It's the proportionality constant between the absorbance and the concentration in moles per liter, or here, or in grams per liter, okay? So you can use this in the Beer's law formula that was mentioned in the lecture about CD that relates absorbance to concentration. Okay, so yeah, so this is a very, very useful thing. It's useful mostly for people who are actually purifying a protein. Oh, and also you can use this to like see if trypsin's if you're gonna do mass spectroscopy or something like that. Anyway, so let's go back to my talk here. So yeah, so we've looked at the gene sequence, okay, at the proteins, at the translation software. So if you've like cloned a piece of DNA, you can use this you can use this translate, XPZ translate. You can just put in the sequence that you sequenced and ask it to translate it. And you can see if there's any mutations in the gene that you actually sequenced. So you can just, like, if you send a plasmid to get sequenced, you get the sequence. There's other softwares that are very sophisticated to do this, but you can just paste it here, ask for it to translate, and it'll translate it for you. Okay, so yeah, so you have the protein sequence, we have the protein parameters. Okay, so now we have the sequence of the protein. We want to say, are there any other proteins with similar sequences in the databases? So this glycogen phosphorylase from rabbit muscle, okay? So we can take the sequence and we can go to another site at the NCBI site, which is called BLAST. Okay, BLAST is here. So it's called a basic local alignment search tool. So you basically get a sequence of DNA or protein, and here there are different modules of it. There's a whole bunch of different modules, okay? But these are the 4 basic ones. You can take a protein sequence, a protein BLAST, and take your protein sequence and see if there's any other proteins with similar sequences, and it's going to align them. It's going to list them in order of the most similar to the least similar, up to a certain cutoff. You can also get nucleotide BLAST, so you can get a DNA sequence or an RNA sequence and look for nucleotide sequences that are similar. And you can also do this BLAST-X and T-BLAST-N, and there's also T-BLAST-X and BLAST-N. Okay, but anyway, BLAST-X takes a nucleotide sequence, it translates it to the protein, and then it looks for protein sequences that are similar to the translated sequence. So it starts with a DNA sequence or an RNA sequence, but it searches a protein database, okay? And then this does the opposite. This T-BLAST-N does the opposite. It takes the protein, and then it takes that protein sequence, and then it compares it to, it looks at the nucleotide database, and it translates everything in all 6 reading frames, and it compares all of those possible sequences to your protein. Okay, so you could use this to like try and find genes, like maybe your protein there's no other protein that's similar to your protein, and you start saying, why is that? Maybe that this gene actually exists in the DNA sequence database, but no one has actually annotated that sequence to be a protein. So you can actually look for sequences similar to your protein in all the nucleotide sequences that independent of their annotation, okay? Anyway, but if we take the glycogen phosphorylase sequence and paste it into this BLAST here, okay? And we go to algorithm parameters, and here, I think the default is 100. So it's going to give me top 100 sequences. But I know, because I've did this already before I came here, I know that there's just so many sequences of this protein. So the first like 50 or 100 proteins are like 99% identical to the glycogen phosphorylase from rabbit, because it's been deposited so many times, and also other mammalian glycogen phosphorylases are very similar. So like the human glycogen phosphorylase is like 95% identical to the rabbit glycogen phosphorylase. So it's not going to be that interesting. So I said, give me the top 1,000 sequences, okay? I could have asked for more, but I was afraid of it crashing, okay? But anyway, so if I go here, let me see if oh no, I go forward, let me see. Yeah, so it basically goes through several rounds of checking, and then it gives me the top 1,000 hits, okay? And it lists them from the top to bottom. So the first hit is actually the actual protein. It comes from the rabbit. This is the muscle rabbit glycogen phosphorylase. But then it's got, and you can see here, percent identity, 100%. And then the next one is also a similar protein, also from rabbit, same name, 99.88%. So there seems to be like probably one amino acid difference or something like that. I don't know why, okay? So anyway, so as you go down, the percentage identity starts dropping, slowly dropping. But you can see the top hits are all from rabbit. But then all of a sudden, now we start seeing other, so this Lepus europaeus, that sounds like another kind of rabbit to me. It's probably some European rabbit species or something like that, okay? And then Scurius, I don't know what that is. Might be a squirrel or something. I'm not sure. I'm not a biologist, so I don't really know. But anyway, as we go down, we go 99%, and then but you can see they're all muscle glycogen phosphorylases, okay? As you keep on going down, Mus ratus norvegicus. That's like a laboratory white rat, Norwegian white rat. These are mice. Mus is some kind of mouse. Okay, so Canis lupus, so dingo. That's like the dingo dog from Australia. Okay, it's some kind of wild dog. Okay, Panthera leo, that's probably a lion. Leopardus, I'm not sure what kind of leopard that is. Okay, so there's a whole bunch of different ones here. And then at pan troglodytes, now we're getting into primates. That's chimpanzee, I think. And then it's a cat, some kind of cat. I'm not sure if we passed humans already. But anyway, so, Homo sapiens here. Muscle form isoform 1, 92% identical. So we've got literally thousands of them. The top ones are all muscle forms, okay? But then as we go down, now we start seeing other things. Well, eventually, I've come down to half my list, and it's still all muscle, okay? And they're all like 88%. And here, Xenopus levis. Does someone know what Xenopus is? It's a laboratory frog. It's a kind of frog. Okay, so this is an amphibian. And its muscle glycogen phosphorylase is 86% identical to ours. Okay? It's a mammalian. So, okay, so keep on going down here. Still muscle, still muscle. This is telling us a lot about evolution, right? Keep on going down. Still muscle. Caramba. Now all of a sudden we start seeing some other things. Brain glycogen phosphorylase. And this is 84% identical. And now it's all of a sudden, it's getting to be mixed of brain and muscle, okay? So you can see that there are like families of proteins in different tissues of ours, has the same protein doing the same kind of thing, but they've evolved separately, sort of two different lineages in the same organism, okay? They've sort of, the gene duplicated, and now one is evolving to do its best activity in muscle, and the other one is probably modifying its activity in the brain. And there's another one in the liver and everything like that. Okay? So as we go down here, most of them now are mostly brain forms. Okay? And these organisms here, I don't recognize them at all. Okay, so anyway, so this is interesting. Trying to find something that I recognize here in the brains. Biologists can raise their voices here. Anyway, so this is 1,000 sequences, and after 1,000 sequences, we still have 83% identity, okay? So there's like literally thousands of these sequences in the genome database, which is it's very good, because there's a lot of information. But when I was your age, it was much more easy to study because you do it and you get like 10 hits or something like that, and that was it. And you had all the information that humanity had about now it's just gotten to be too much. But anyway. So yeah, so what I can do, so now what I might want to be interested in doing I want to get some kind of information about what sequences, what parts of the protein are more conserved than the others. Now in this case, this is not a good example because 1,000 proteins, and they're all at least 80% identical with each other. Like the worst here is 83% identical with the first one. So they're all very similar. It would be more interesting if the ones at the bottom were like 25% identical or something like that, then I could get more information. So I'd have to get more sequences here, or I would have to start eliminating. There are ways to eliminate the redundancy. I could say, eliminate all sequences that are equal to other sequences, and just give me one, or something like that. You know, there's a whole bunch of different ways to do that, but I'm not going to do that now. But what I can do, I can just basically select. Here, there's selected 1,000 sequences. I can unselect them and then just start selecting sequences that are different in their identities. So I have 100%, and then I'll just go down to say 95, 90, 97, 96%, 95%. Let me see, 95%. 95% here. And then I go down to 90%, say. Go to 90%. And then I go to 86%. I could choose a whole bunch of different things. Okay, then 83%. Then there's a whole bunch of them with 83%, it seems. Anyway, get this guy here. Okay, so I could choose just these ones, and I can just choose them, and I could get their FASTA files. Okay, I could do a multiple sequence alignment. Okay, I can download their sequences in FASTA sequence, okay? For example, this, if I take this here, then here, if I open it up, I've got their FASTA sequences here. So one protein, then another protein, then another protein, et cetera, all the way down, okay? The ones that I selected. If I'd selected all of them, I would get like a text file with 1,000 sequences. It'd be really big, okay? But then I can go here, I can get those sequences, and I can align them. Now I did this before I came here, but I just go multiple alignments here. And now, so this was an alignment that I did before, and now it's going to do it again with those sequences that I chose. Okay? I chose fewer sequences, but let's see what's going on here. Okay, so now it aligned the sequences. So I've got amino acids 1 to 843, and it's basically the red areas are where the sequences are very similar, and there's these blue lines here. It seems like there are places where the sequences are a little different, but it seems like most of it is very, very similar, which is what we were expecting. But what I can do, I can zoom in on the sequence. I can take this here and zoom in. I thought I could zoom in. Hello? I thought I could zoom in. This doesn't seem to be showing anything for some reason. I think something went wrong with this thing here. So I'm going to go back. I shouldn't have gone back. I probably lost here. Okay, so coloring. RAS small amino acid coloring. Yeah. I don't know why. Here we go. Okay, yeah. So now it's coloring the amino acids according to their properties. Okay, so I'm not sure, I don't know what amino acid is each color, but if I go further in, I can start zooming in and we'll start, now we start seeing the sequences of the amino acids. So V is green, L is green. So these hydrophobic amino acids are green. E is glutamate. So acidic residues are red. Basic residues are blue. And then we have light blue, which are these glutamines and asparagines that are polar non-charged residues. Methionine and cysteine that have sulfur atoms, etc. Threonine and serine are orange. They have hydroxyl side groups. Okay, so they are this coloring. You can choose different coloring schemes here to sort of like let you have an idea of what is being conserved or not. And then you can see these positions here where you have, I'm going to zoom in a little more so you can see it maybe better, or I can see it a little better. Okay, you can see here like this position here, the valine is conserved in all of these proteins. Okay, but for example, this asparagine in these muscle proteins, these are probably brain proteins. They seem to be changed to aspartate for some reason. Might be just genetic drift, or there might be some kind of importance for this change here, okay? And then you can see here, this is a glutamate, no, this is a glutamine, and this is changing to glutamate, okay? And this is valine changing to cysteine. So this is a more drastic change, okay? So you can like, and here, we're in this part of the sequence, oops, we can go back and forth. Okay, so you can like go along the sequence to see, and I think this is like the conservation profile, these bars here. Okay, so anyway, so you can look at this. There are a whole bunch of other different programs that allow you to do this kind of alignment if you want. Okay, so next. Okay, so I can get these sequences, and if I want, I can take them to another program that allows me to align the sequences in a more controlled way. And so there's this cluster multiple sequence alignment site, okay? So that has a whole bunch of different, oh, I shouldn't have done that. Okay, so this cluster omega site is a multiple sequence alignment site. So I can paste in the sequences in FASTA format, okay, of glycogen phosphorylase from rabbit, and then from, oops, and then from this dingo dog. This is what I did before, the dog. And then this one here is from the brain from the cat, okay? So we're aligning all these different ones, okay? And then another one here is Xenopus. This is the frog, okay? So there's a whole bunch of different ones here, and we ask it to align, and it will align the sequences, and it will output in a whole bunch of different formats, okay? Phylogenetic tree, etc. Okay, there's a whole bunch of different ways. But the most basic way is this, just basically aligning them with their code, with their deposition codes here. This comes from the first line of the FASTA file. You can change this. So I could have written in rabbit muscle here and cat muscle here and then Xenopus brain here or something like that. Okay? So here, basically, it's aligning them. And you can see at the very bottom, there's a line with asterisks, which say that all of the proteins have the same amino acid at that position, if there's an asterisk here. If there's two dots, if there's like a colon, then it says that at this position, there are amino acids that are very similar to each other. So here it seems to be lysines and arginines. Arginines and lysines are both basic amino acids, positively charged amino acids. So these are conservative mutations, okay, or conservative differences. But then you can have this, like where there's a gap here. That means that there's differences that are considered to be very drastic differences. So here we have a valine and a glutamate, whereas up here at the top, we have a leucine and a glycine. So that's very, very different. It goes from a small amino acid to a large amino acid, and a negatively charged amino acid to a non-polar amino acid, a very, very small non-polar amino acid. This is a very big change here, okay? Now, it might be difficult to look at this. So at the very bottom here, you can say alignment with colors show, and it will show you the same thing, but now you have the amino acids shown in their colors coding for their properties. So like I said, charged amino acids are blue or red, etc. Okay? Yeah, so this cluster is very, 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 very interesting to use. Now, if I'm interested to see if the structure of the protein is available in the databases. So I can go to the protein data bank, the PDB. Okay, so this is the site here, the PDB, and I can look up. Let me see if I could look it up here. Yeah, if I look up here, if I just put in rabbit muscle glycogen phosphorylase, I'll get a whole bunch of hits, okay? I'll get a whole bunch of hits. One of which, sorry, well, I'm going to show a whole bunch of hits here. Hold on here. This is one of them. So yeah, so this protein data bank, it has all of the sequences, all of the proteins whose structures were determined by NMR, crystallography, and cryo-EM. I don't think it has the, it's only experimental models. It doesn't have the, oh, it's got 1 million computed structures. It's also got 1 million computed structures. So it seems to be linked to the, probably linked to the AlphaFold database, because there's 1 million computed structures here, okay? But there's also 234,000 deposited structures, experimentally determined structures, okay? And there's a whole bunch of interesting educational information here. You can explore this a lot, okay? But if you put in rabbit muscle, glycogen, phosphorylase, what happened here? Why didn't it find a whole bunch? What's that? I didn't understand what you said. Huh? I don't think I'm hearing you guys. What are you saying? Oh, there's a typo. Glycogen phosphoryl. Wow, it doesn't even, there's no artificial intelligence on this site. Ah, there we go. Okay. So there's a whole bunch of structures of rabbit glycogen phosphorylase. It's been studied a lot. Okay? And yeah, so there's a whole bunch of them, and it's been determined in the presence of a whole bunch of different cofactors. Remember, I said it has pyridoxal phosphate as a cofactor in the active site. So it's been crystallized in a whole bunch of different states, and so you have a whole bunch of different depositions, okay, by different groups, and you can choose the one you want. And it has also, and many of them will have the reference in the literature where you can find where the structure was described in the scientific literature. So you can download the paper, okay? So if you just choose any one of these, okay, let's just choose the first one here. I had chosen another one, but I probably lost the link. We'll just choose this one here, okay? So this is the structure in complex with a potential drug, hypoglycemic drug, and it's also in complex with pyridoxal phosphate. PLP is a pyridoxal phosphate. So if I click on this 1LWN, I will go to the page of that deposition, okay? So now here, there's a whole bunch of information, interesting information. First, it'll tell me who are the authors. Was there a paper? There was a paper where it was described initially in Biochimica Biophysica Acta, okay, in 2003. So I can download it, clicking on this DOI. I'll probably go to the PubMed deposition, okay? Now, there's some kind of good information here. So it's 2 angstrom resolution, okay? So this is telling me something about the quality of the data set that was used for the structure. This R-free, this R value, there's an R-free and an R-work. This is interesting to tell you what it is. Remember I told you that the X-ray diffraction pattern, you get the X-ray, there's a whole bunch of different reflections, and they all have different intensities. There's some that are much more intense than others, okay? So basically the data set is a text file where every reflection has HKL, every reflection has an index, a position, and it's got the intensity or the structure factor, which is the square root of the intensity, I think. Yeah, it's the square root of the intensity. So anyway, so it's going to give you the structure factor, which is like the amplitude of the wave, okay? And you can calculate, so this is the FOBS. This is experimental. Now, they've deposited a structure, and from the structure, you can do the Fourier transform of the structure and calculate what the theoretical diffraction pattern should be. It doesn't make the image, it just has a table of all of the theoretical amplitudes of each of the reflections. Okay, so you'll have, like, say, for HKL 6, 8, 15. Let's say, so there's the coordinates of that particular reflection, and this FOBS is, let's say, 1053. But you could also calculate what the FOBS is, calculated, okay, for that reflection, and say it is 850. Okay. And you can do this for all thousands of all the reflections. And then, so what you do is you calculate what they call an R factor, which is the sum of the FOBS minus F calc, and it's the absolute values of them. And it's the absolute values of the differences between the absolute values divided by the sum of the absolute values of the FOBS. Okay, so this is basically giving you the average difference between the observed data set and the theoretical diffraction pattern of the model that was deposited. So you want this number to be as small as possible. Okay, and here you can see this number is not bad. It's 0.218. So that's basically saying on average, the model gives a diffraction pattern that's 20% identical or 20% different. The difference is only 20% from that of the experimental data set, okay? So this is basically saying that the model is fitting relatively well the data, okay? Now, what this here says, so you can fit a model to be perfect with the data, okay? The thing is, you also have to be chemically realistic. So when your model has to have carbon-carbon bonds that have the right lengths. It has to have torsion angles that are considered realistic. It has to have phi and psi combinations that fit into the that are allowed in the Ramachandran plot. It can't have atoms too close to each other that have their van der Waals volumes superposing, because that would have a lot of repulsions. So there's a whole bunch of metrics that you'd use to look at the quality, the chemical quality of your model, okay? So you have to balance the two things. You have to have a model that fits your data as good as possible, but is still chemically reasonable. And this here on the right-hand side is saying how good the model is in comparison with other structures in the database that are in the same resolution range. So other proteins that were deposited, that have resolutions around 2 angstroms, this clash score tells about whether the chemical groups are not clashing too much in the model, okay? So it seems to be, on average, it's not bad. Ramachandran outliers, 0.2%. So 0.2% of the amino acids in the protein are in non-allowed Ramachandran regions, okay? That's not bad. 0.2% is not that bad, and it's sort of like in the middle. That's okay. And then you have side chain outliers. The side chains, so the side chains of the amino acids, you can statistically look at the configurations of side chains of each amino acid in well-defined X-ray structures and say, let's say arginine has 10 different rotamers that are very, very common. And serine has 3. And valine has 3, etc. And you can see the percentage of these. You can say, well, in arginine, there's one that's all 15%, and the other one's 10%, da, 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 da. But there's, and then you can say, well, in this structure, are there side chains that are adopting conformations that have never been seen before, or hardly ever seen before? And that will rank them. And here it seems that the side chain outliers, it seems like they're using a lot of side chains. A lot of their amino acids have side chains and configurations that are not that common, okay? So you might sort of like think about, well, why is that, you know? So it's good to sort of at least appreciate this so that when you actually look at the model, so you can download the structure. So now you can download the structure, okay? So you can go here, download files. You can download 2 different things. You can download the PDB file. We'll download that. And you can also download another file, which is called the MTZ file. Let me see here. Doesn't seem to have the MTZ file here for some reason. It's got this MCIF file here that might be enough to give me the, this is probably just going to, yeah, this is probably going to give me the, I'm not sure if this is going to give me the structure factors. But anyway, let's look at the PDB file. So we can like display the PDB file here, okay? And you can see what the PDB file is. So this PDB file is a text file that has some information about the structure determination. And then as we go down here, we can see, oops, yeah. So as we see here, it starts listing the atoms and the positions of the atoms in the PDB file. So we have serine serine number 5. The first 4 amino acids seem to have been lost. For some reason, they were not visible in the electron density map, so the model does not have them for some reason. Okay? You'd have to read the paper to find out why. Okay? But now we're looking at serine number 5, and we're looking at the nitrogen, the alpha carbon, the carbon of the carbonyl group, then the oxygen atom, and then the beta carbon, and then the gamma oxygen. So we have serine, the nitrogen, the carbon, alpha carbon, the carbon, the oxygen, and then, so always the first 4 atoms in the PDB file are these 4, NCACO. And then they will describe the side chain. In the case of serine, there's the beta carbon and oxygen, which is in the gamma position, because alpha, beta, gamma, delta, epsilon, etc. Okay? Here, the hydrogens are not shown because at this resolution, 2 angstrom resolution, you don't see hydrogens. Okay? So the hydrogens were not shown. If it was an NMR structure, the hydrogens would be shown, because NMR, the structure depends on nuclear Overhauser effect distances between hydrogen atoms. So you actually know the positions, well, at least the models show the positions of the hydrogens, okay? But X-ray crystallography, you don't see hydrogens unless it's a very, very high resolution structure, and then you can see the hydrogens and they will be put in. But hardly ever they'll put in the hydrogens, okay? And then we have aspartate number 6, and we have the nitrogen, carbon alpha, oxygen, carbon beta, carbon gamma, and then 2 oxygen deltas, because there's a carboxylate group there, so there's 2 oxygens at the delta position. And these are the positions, the X, Y, Z positions of the atoms in the unit cell, okay, in the repeating unit of the crystal, okay? And this here is the occupancy, which is saying that they've calculated that that atom is always at that position 100% of the time, okay? Now, there are some times that you can find a serine, or you can find a certain amino acid in the, you can see sometimes that the oxygen is in this direction, and then you can see also the oxygen in this direction. So there's like you know it's a serine, but the electron density is like this, let's say, okay? And you say, well, how can that be? It means that my serine is sometimes this pointing this way, and sometimes the oxygen is here. So what you'll do is you'll have 2 different lines for the oxygen. You'll have one line for the oxygen 50% of the time or 70% of the time in one direction, and 50% of the time in the other direction, or something like that. So this is the occupancy, they say. And this is the B factor. This B factor is called the temperature factor, or the B factor. It's a parameter that determines how well defined is the position of the atom at that spot. So it's basically, if it's a high number, it's as if the temperature is high and the molecule is vibrating a lot around that position. So it's more spread out around. So if it's a low number, it's as if that atom is very, very well defined at that position. And if it's a higher number, it's because it's not very well defined. Now, there's many, many reasons that that number could be high. It could be that really it's very badly defined. It could be very, very flexible, even though it's in the crystal. But it could be because the data is not good, or the model is not good, or something like that. There's a whole bunch of different reasons for a temperature factor being high or not. But it is one of the parameters that are used to fit the data, okay? So it's just for you to understand what this is, okay? Okay, so anyway, so you can go, you can download this file, and you can open it in PyMol, or in another crystallographic molecular graphics program and analyze it just like I analyzed hemoglobin, okay? I'm not going to do that because we don't have time, okay? So I'm going to end with my last couple of slides, I think. Where was I? Okay, I passed through all of this. Okay, so now the last couple of things I want to just talk about protein modeling guided by coevolution data. So this is going to lead up to the AlphaFold story, okay? Just a little introduction on it, and then you're going to talk about it a lot in the other lectures. So there are, like I showed in the other slide, there are on the order of magnitude of a couple of 100,000 experimentally determined structures during the last 50 years, okay, in the databases. But because of huge sequencing projects, mostly from metagenome projects. So nowadays, we have sequencers that are really, really cheap. You can go and get seawater or soil in your backyard or vegetable matter, purify the DNA, put it in a sequencer, and it will sequence, it'll give you millions of reads or billions of reads, okay? And you just deposit that into the databases. And it might be sequencing a whole bunch of different organisms at the same time. But the thing is, there are stretches of sequences, and if you translate them, you realize that it's coding for a protein, and you compare that protein. Well, it could be coding for a protein, you don't know, but then you compare it with the sequences, all the other sequences in the databases, and you see that that sequence, the protein that's being coded by that sequence, is showing up. You can align it just like we aligned in the BLAST algorithm. So now we've got billions of sequences of proteins, and that allows us to make a whole bunch of why am I sitting down? A whole bunch of superfamilies of homologs, okay? So you can basically, you have a very, very deep vision of the relationships between certain proteins and other proteins. So in the past, when we had fewer sequences, we would have a structure of a protein and another structure of a protein, and they seem to have the same fold, but the sequences are very, very different. So it's sort of like anthropology or archaeology when we see, like, you know, a mouse skeleton and a human skeleton, and we don't know if there's any link. And then we start finding other skeletons of other animals that are sort of in the middle, and we start being able to make a link between them. Okay? That wasn't a very good example, but anyway, you know what I mean. So now we have so much sequence data, so we can actually find relationships between distant sequences that seem to be very, very distant in the past, but now we can actually see that they're all related to each other. So this has helped a lot in the protein structure prediction field, okay? I mentioned this critical assessment of protein structure prediction. This is a contest that's been going on since like the 1990s or something like that, at least, I think, where structural biologists would determine the structure of a protein, but they wouldn't publish it. They wouldn't publicize it, but they would send it to the organizers of this meeting, and every couple of years, people who were specialists in predicting the structure of proteins would take the sequence and they'd try and predict the structure, and then they'd compare their predicted structure, their model, with the actual structure. And until like maybe 10 years ago, this was not a very, very good. It wasn't very successful. It was getting to be good for small proteins, up to say 80 amino acids or something like that, but it wasn't very good for bigger proteins, and even for some small proteins, it wasn't very, you'd have to have a lot of, well, I don't know. It just wasn't that successful. It wasn't very, very satisfying. The success rate was not very good. But then all of a sudden, in the last like 10 years, especially in the last five years or 6 years, two groups, actually several groups, but led by the David Baker group at the University of Washington, and then by the DeepMind group that had the computation power of Google, were able to use this huge amount of sequence data, metagenomic sequence data information, to help them to predict the structures of proteins. So this is David Baker, and this is Demis Hassabis and John Jumper that worked at Google. Okay, so these guys did the AlphaFold, and these guys did Rosetta and Robetta. Okay, so they published several papers over the last less than 10 years. This came out in 2017. Okay, so protein structure determination using metagenome sequence data. So these guys said that we integrate metagenome data, contact-based structure matching, and Rosetta structure calculations to generate models for 614 proteins, protein families with currently unknown structures. 206 are membrane proteins, 137 have folds not represented in the protein data bank. Okay, so these guys were showing that you could use these alignments to somehow improve the quality of the structure predictions. Okay? So what's the basis of this? The basis of this is based on an idea that was actually much older. It was called statistical coupling analysis, or SCA. Statistical. Okay, so basically, that said that if I take an alignment of protein sequences, and I can look at every position in the alignment, and I can say, when this amino acid is a certain amino acid, how does that correlate with the amino acids at every other position in the protein? And is there some kind of correlation? Is there some kind of like co-evolution going on? Okay, for example, here you can see this orange one here. When it's orange, this is green. And when this is green, this is orange. So there seems to be a correlation between these two positions. Okay, whereas there's no correlation between, say, this position and this position here, because this is orange, this is blue, this is orange, this is orange, this is green, this is yellow, this is green, this is blue. It's not very much correlation. So this kind of correlation, okay, can be analyzed statistically. If you have a large enough alignment, and this large alignment is only available in the last 10 years, say, because of this metagenome sequencing, okay, it allows you to get statistics that are reliable to use in predictions, okay? So you can do this, and that allows you to say, okay, well, that means I can now, when I do a modeling, when I try and determine the models, I can say, if there's correlation between two positions in the sequence, evolutionarily, that might mean, doesn't necessarily mean, but it might mean that those two amino acids are interacting with each other, either directly or indirectly, but they're probably close to each other in the structure. So I can use that to guide my protein modeling algorithm, okay? So you can do this. So you can basically have all of these correlations between different positions. And this actually looks a lot like those NMR-based studies where you had NOEs, where you had like this position has hydrogen atoms that are close to these hydrogen atoms, et cetera. It's the same kind of information, but the NMR experiment it's coming from a different place, right? NMR, it's in an NMR tube. Here it's from evolution, okay? But it's very similar at this general level, okay? And that allows you to make a model that is consistent with these correlations, okay? So yeah, so then AlphaFold also in 2021, okay, they said, so this is this CASP 14. So now they were, these guys in CASP, the Rosetta group and the AlphaFold group, they were all of a sudden in this CASP 14, the 14th CASP challenge, they just blew everyone else out of the water, okay? They were able to predict like a huge number of structures relatively well, okay? And then the DeepMind and everything like that. Then these guys started, you know, depositing 350,000 structures, okay? And now they've deposited over 1 million, okay? So anyway, yeah, so I'm just going through all these guys here. So they got the Nobel Prize last year. And I just wanted to show one last thing here. So you can, there are two main sites. You can download this and put it on your workstation or your server. But you can, there are two sites. There's an AlphaFold server, I'm not going to talk about that. But you can, there's a protein structure database that has, I'm not sure how many million sequences in it or structures in it. But you can just basically put your, you can just put the name of your structure or the UniProt accession number or something like that, or the gene code or something like that, and it will try and find a model for that structure, okay? So if the sequence is there, and it will give you the model, their model for it, okay? So here I'm gonna just show you one model of a protein that we're studying, okay? It doesn't really matter what the protein is, but you can, it basically gives you something like this. And what do the colors mean? The blue colors, if they're blue, then the quality of this prediction, they say, is very high. If it's red or yellow or orange, then the quality is not very good. If it's light blue, it's sort of intermediate. So you can see here that there seems to be a relatively good, a well-defined domain here. There seems to be a central domain here that's not very well-defined, and a C-terminal domain here that has a relatively well-defined central core, but the rest of it is not very well-defined. And then there's this big loop here that connects this last domain with this second last domain here. And here, and this is an interesting thing here, this map here is called a predicted alignment error map, okay? So basically what does it show? It shows, it's this, remember, this protein has around 850 amino acids. So it goes from amino acid 0 to 800, and here's 0 to 800, okay? So it's basically saying in this model, what's the confidence that that atom, in the model, that atom, or that residue is close to the other atoms. So for example, in this region here, the first 200 amino acids, the quality, the expected error is low because it's dark green. So dark green is good here, okay? So the first 200 amino acids here correspond actually to this domain here, okay? And it's saying that the quality of this structure for this amino terminus domain is relatively good because the first 200 amino acids, the expected error of them internally, among themselves, is relatively good. But their relative position in comparison with the rest of the protein is terrible because here, it's very, very high. Or here, you go in this direction, it's very, very high, okay? Except for maybe a few amino acids here, okay? And then they have a central domain, which is this here, okay? Whose quality doesn't seem to be very good. There seems to be some little parts, little stretches that are relatively good, okay? But in overall, it's not that good of a quality. And that's why these colors here are sort of orange and light blue. Okay? And then we have a C-terminal domain that's relatively well-defined. The last 100 amino acids are relatively well-defined among themselves, which is this part here. But it, in respect to the rest of the molecule, is very, very badly defined. Okay? So basically, this is saying to me, what can I believe here and what can't I believe? I can believe in the first, maybe in the structure of the first domain, maybe in the structure of a part of the last domain. I don't know how these are oriented with respect to each other. And this central domain, not really sure how much I want to believe that. Okay? So that's this kind of interesting type of thing. Okay? So that's where I wanted to, oh yeah, so this is what I wanted to end. I don't know if you guys want to give like exercises to the students. Okay? I'm not sure if I'm gonna, I can make available my talk. And then like at the end of it, we'll have this here. So if you want to do, you might have, maybe all of you have already done this, but it might be trivial. But some people, if they want to do it, they can. Install PyMol or Chimera X on your computer. So these are molecular graphics programs, okay? And these are the sites where you can download them. Choose a protein of your interest and search for it or close homologs in the protein data bank. So this is the site for the protein data bank. And then answer these questions. It's basically just saying, okay, put the model in the program and try and look at, interesting parts of the structure. Amino acids, or like, highlight certain structures with different colors and everything. Just learn how to use the program, okay? That's basically what I'm asking you to do. It's a very, very simple thing. Maybe a lot of you have already done it before, okay? And that's it, end of part 3. Questions? Everyone wants to go home. Explore the bars in Villa Madalena or something like that. Yeah.
With respect with B factors and vibration, is there some direct relation with flexibility?
Yes.
And also with the resolution map of cryo-EM?
Yeah, well, the B factors have to do with flexibility. If the molecule is very flexible, in fact, in a cryo-EM map or in a crystallography map, if it's very flexible, you just will not see any of the electron density for that. And if you try and model it, it will give you a high B factor because it's not very well defined. Cryo-EM maps also, I think, they also model the B factors, but I can't really comment on the technical aspects of that. Yeah. Okay, so hopefully this has been at least a useful revision about protein structure on the first day, and this will allow you to appreciate the more technical and more detailed exercises they're going to do in the next couple of days in this course. Okay? Yep.